I'm Alon Diamant Carmel. I'm a researcher at the MyHeritage science team, and I'm really excited to be here with you today and to talk to you about some of the things that we're cooking that will come up in the next month. And the topic of my presentation is going to be the worldwide DNA web, or in its alternative title, genetic groups. So, as you probably heard yesterday in Gilad's keynote announcements, MyHeritage has been working on improvements to its ethnicity estimation models. And I will be talking today on one research direction that we at the science team have been focused on: genetic groups. Some of them Gilad demonstrated in his keynote talk yesterday. I will explain how we detect such groups using billions of relative matches, such as the ones that you all receive in your reports. How we construct a worldwide network of matches spanning millions of users. And then I'll tell you about some of the interesting groups that we were able to detect and have been researching in the past few months. So let's begin with a very brief introduction on what are matches, what are their typical size, and our sensitivity and ability to detect them. Relative matching is at the core of genetic genealogy. Using relative matching, we at MyHeritage have gathered hundreds of success stories connecting individuals like Holocaust survivors, siblings that were separated after birth, adoptees searching for their biological parents. These individual success stories of families reunited are one of the most rewarding things, whether they happen to users across the world or to friends of ours at the science team. So what is an IBD match? A match is a long stretch of DNA that two individuals share. If this DNA segment was descended from a common ancestor, we say that it is identical by descent, or IBD. Now let's examine briefly a few examples for typical matches. Frank and Molly have two children, Ben and Lisa. Ben and Lisa each have a set of chromosomes from Frank and a set of chromosomes from Molly. Ben's paternal chromosome, marked here in purple, is a random mix, a recombination of two of Frank's chromosomes. At the same time, Lisa's sister has a paternal copy of chromosomes that is also the result of random recombination. Now let's look at the identical segments by descent that they both share. Okay. The marked segments on the right are identical because here both Ben and Lisa inherited the same regions of Frank's chromosomes. And the same goes for copies of chromosomes that they inherited from their mother Molly. All these segments are identical by descent. When Ben and Lisa married and had their own children, each of the two cousins again received a recombinant chromosome. The shared parts are the ones that they were lucky enough both to inherit from Frank and Molly through their parents, Ben and Lisa. After 2 rounds of recombination. For individuals like Ben and Lisa, two siblings, the average shared IBD between them is 50% of their genome. Lisa's son, however, will typically have a 25% match with Ben, his uncle. Well, the two cousins will share only 12.5% of their DNA. Each recombination event along the path to their common ancestor divides the average match that we expect to see by 2. So if we look at a path of 3 recombination, from me, from a person, individual, we have an average size of 25% for a match, either with my uncle or with my nephew. 4 recombinations away, we have a typical match of 12.5%. 5 recombinations away, it goes down to 6%. 6 recombinations away, it's 3%, and so on. By the time we get to 5th cousins, 12 recombinations away from one another, they are related, assuming they are related only from one side of the family, they will have, on average, only 0.05% of shared segments between them. Their common ancestors would have lived 6 generations ago, less than 200 years ago. This graph summarizes the journey that we just did. And it shows the typical expected size of an IBD match, depending on the number of generations ago that our common ancestors lived. And we can see here the 5th cousins. So this is what we expect to see on average. And this is an important word, because every recombination is random and unique. And the particular series of recombinations along the path from me to the person I am matched to will determine the actual IBD that we will find. And we may ask ourselves the following question. What is the probability that we will be so unlucky as to not to find any IBD between 2 relatives? That they will have, the 2 relatives will have 0 segments in common. That somehow, through repeated recombinations, they inherited totally different DNA regions from their common ancestors. So, through careful simulations, scientists have come up with a model that tries to answer this question. And the result is before you, in this graph. This graph shows what are the chances for an event where 2 relatives do not share any IBD match between them, depending on the number of generations from their common ancestor. And we can observe that the chances become very, very slim once we get to 7th or 8th cousin here. To conclude what I said so far, with some probability, 2 related individuals may not share any DNA segments. The size and the probability of finding shared segments decrease as individuals become more distant. And typically, we can learn about our history from IBD matches as far back as about 8 to 10 generations, because otherwise the probability of a match becomes very, very small. So now let us move from individual matches and family reunions to billions of matches spanning worldwide populations. We will try to imagine a worldwide network of matches and its characteristics, beginning with all the matches of a single individual. All of you here probably have thousands of matches in your MyHeritage accounts. Too many for me to draw on a single slide. So let's imagine just a few of these matches here in this tiny web that we're weaving on this slide, with a single user at the center. Now, let's add a second round of matches between all the matches, between all the relatives of that user, that you see here. But let's not forget that many of them also have matches between them that I just added to this slide. And our web is becoming more thick and complex. See how densely connected, closely related individuals are. Like this family here. Let's remember this picture of this family. And let's try to answer how will this web look like for a lot more matches. Many studies in the literature have tried to generate larger networks of IBD matches, anywhere between about 2,000 individuals to 20,000 individuals. These networks have been used to study population structure, but have also been extended to other organisms, like the last article here, last research here, that studies the relations between different pathogens using IBD networks and tries to understand the mechanisms of disease using such networks. So let's draw one such network, for example, with a few thousand individuals on it. On the right-hand side, you can see an example where individuals are colored according to their region of origin. And we can see that many of the blue individuals, for instance, form dense structures, and they stay closely related. Taking all of the matches that we have in the MyHeritage dataset generates a massive network containing billions of connections of matches between people. For instance, to generate the genetic groups that I'm going to show you, we used over 1.5 million individuals and over 10 billion matches. Trying to visualize such a huge network is quite tricky, but we can borrow from studies on social networks and communication networks. And the following slide is what part of the World Wide Web, the internet, looked like when it was visualized a few years ago. Again, see how some parts of this web tend to be more dense than others and have many connections between them. In these examples, we saw the patterns that are typical of many networks, including IBD matches. These denser regions are called clusters. And they may represent, when looking at the smaller scale, families, for example. However, at the larger scale, they may represent populations. At the population level, any given pair of individuals is related through many common, however distant, ancestors. For a pair of individuals on different continents, the relationships may be too distant to result in any detectable IBD. However, pairs of individuals from the same geographic region may have many recent common ancestors. On the individual level, as I showed you, not every relationship results in IBD sharing. But on the population level, when we have many people in a group, in a genetically related group, matches are more frequent when they have many common recent ancestors. There is no clear and exact definition of what a cluster is. Intuitively or qualitatively, we look for the following properties. We want to have dense connections between individuals in the same clusters. We want to have few connections between individuals in different clusters. However, mathematically, there are dozens and more ways to try to represent these properties. In addition, detecting where one cluster ends and another cluster begins can be sometimes hard. So it's a very interesting challenge to try to detect these structures in large networks. Indeed, many algorithms exist that try to detect clusters in large networks like the DNA web that I just showed you. We applied some of these algorithms to detect clusters in the MyHeritage DNA web, and we call them genetic groups. They come in different shapes and sizes and contain many riddles that we are trying to figure out. Who are these closely related people, and what does their grouping together represent? So now, let's have a look at some of the interesting groups that we found in our network. I'll show you 8 examples of groups, where each example demonstrates one or more of the properties listed here on this slide. Throughout this part of the talk, I will also show you some of the tools that we developed to study genetic groups. These include, for instance, different types of data that we collect, summarize, and display. So this is the first example that demonstrates the surprising resolution that groups may reach. In this group, the distribution of people, geographically, and their ancestors throughout the years almost never extends beyond a very tight and neat region in Finland, shown here on the map. In addition, when we look at the ethnicity of these individuals, the vast majority of them receive high percentages of Finnish ancestry. Looking at the region map of Finland, there is little doubt that this genetic group represents the population that resides in the Pirkanmaa region. And I hope I said that correctly, if anyone can verify. Ah, welcome. And we have more similar groups like this one for other regions in Finland. By the way, each of you is invited, once I reveal a group, to just stand up and confess. In fact, when we overlay multiple European groups, one next to the other, we often see that they hardly overlap, like these three different groups that I painted here on the map. Despite people occasionally moving from place to place, these populations remained isolated enough that we can detect this in our matching network. These groups are relatively similar in terms of their ethnicities. The same 4 ethnicities that you see here above for each group dominate their distribution. Surprisingly, the North and West European ethnicity increases as we go north and west. And at the same time, East European ethnicities slightly increase as we go east and south. This happens across only a few hundred kilometers. So this is another example for a high-resolution cluster. This French genetic group comes from a very specific region in the country. And even more interestingly, their ethnicity is not what you'd expect from a French population. They are hardly Northwest European or Iberian. Rather, they are mostly Irish, Scottish, and Welsh. So what is going on here? So these are the Bretons. They migrated to the region between the 3rd and the 9th century. They share origins with Scottish, Welsh, and even Galician Spanish populations. You can see here the map of their migration from the 6th century. They are part of the family of Celtic people, and specifically the Brittonic speaking people. Their traditional language is Breton, and by now, today, only about 20% of them still speak Breton. But they have their distinct culture and their region of where they reside. And you can see here, for example, a pipe band that is typical of this region and influenced by Scottish music. For the next genetic group, I would like for us to have a look at a new type of data. This is a list of surnames here on the left that is typical of a single genetic group. On the left-hand side are names and how frequently they appear in family trees in this group. On the right-hand side, we show how often they tend to appear in different countries. What is their story? They appear to be Canadian. This is the country where they are most common. Of French ethnicity, perhaps, because they are also popular in France. However, we also see that these names are quite common in the USA, almost as much as they do in Canada. So let's have a closer look at the documented birth and death locations of ancestors in this group and try to figure this out. We put a lot of effort into thinking of ways to visualize the data in order to understand it better. And I'll introduce some additional plots to you during this talk. You may have seen this graph yesterday at Gilad's keynote. This single graph represents a major part of the geographical history of this genetic group throughout generations of ancestors in the past 200 years. Let's see how it does that. This graph shows us flows of people. People coming out of countries, people coming into countries, and people staying right where they were born. It is divided into periods of 50 years, half of centuries. People are moving from left to right, and the colors represent their country of origin. Let's look at one flow, for example. Its orange color here, this flow, means that the movement or migration of people are from here is from Canada. So they started their lives here in Canada and moved here to the United States. In the next half of century, these people born in the United States stayed in the States. And therefore, they're flowing into the blue region from here to here. All in all, we see in this graph two major migrations in this group of people coming from Canada into the United States. The width of each flow is proportional to the number of ancestors that we know about that took part in this migration. And therefore, there is a strong presence here of people in the United States, as you see from this width of flow. Also, there are no significant flows out of the USA into Canada. So this relation is not symmetric. Now let's see these events on a map. The animation on the right shows these migration events. Indeed, people that were in the Montreal area in 1800 flowed in the second half of the century in great numbers into New England and Massachusetts. And it appears that most of them stayed there to this day. These are the French-Canadian Americans. There are actually about 2 million US citizens today that are of French-Canadian descent, which is quite a lot. They immigrated to New England between 1840 and 1930, very close to what we saw in the flow graphs. They worked in sawmills and in textile, as you can see in this historic picture. And they tended to preserve their culture more than other ethnicities that migrated into the USA. Many of them speak French at home. They remained Catholic. And they formed, for a while at least, Little Canada neighborhoods throughout New England. Aided with these new tools that I showed you for studying migration, let's see another one interesting story of migration. On the right-hand side, you can see the distribution of DNA kits today, which seems to be all over the place. From Central Europe to Siberia, to Kazakhstan, to Uzbekistan. And I can tell you that some are even in the USA too. On the left-hand side, you can see something strange about this group. Since it has a strong Northwest European ethnicity that is quite different from its surrounding population. However, If we play again the locations of ancestors of this group on the map here, you can see the year 1700, 1750, and so on. We see that in the 1700s, they all lived, as far as we know, in Germany. And they moved quite quickly to the Russian Empire in the second half of the 18th century. They left the region almost completely 200 years later. And now they are scattered all over. The flow diagram tells us the same story in a single frame. We see people flowing into Russia until the end of the first half of the 19th century. And then strong flows out within the Soviet Union and out of the Soviet Union during the 20th century. What's interesting is that on one hand, they are a distinct group from other German populations that we see. On the other hand, they form a distinct group from Russian populations that we see. And that period of time, these 200 years, was enough to define them as a genetic group. These are the Volga Germans. About 600,000 people today, Volga Germans, live in Russia, Kazakhstan, Kyrgyzstan, and Uzbekistan. They were invited in the late 18th century by Catherine the Great to come and establish farming settlements in the Russian Empire, including the Volga region, among others. And they preserved their culture. They kept their German language, their traditions, their churches, and even their surnames. During World War II, however, about 1 million of them were forced to exile in the Soviet Union. Some of them were sent to work camps, and very little from these populations stayed in this Volga region. While researching this, I discovered that one of my favorite 20th century composers, Alfred Schnittke, is in fact part of this group. His father, a German, came to the Volga region at a later stage than the main migration flow and married his Volga German mother, demonstrating the cultural ties that remained between the settlements in Russia and their country of origin. The following group is very similar to the Volga Germans. It also describes migration, but what's interesting is that the genetic groups are able to differentiate between these two stories, even though they are quite similar. So let's see. Again, these are Northwest European people, this time in Ukraine, in the region of Odessa, next to the Black Sea. We see here that they also left Germany, but about 50 years later than the Volga Germans. They came from southwest Germany and Alsace, and moved to the part of the Russian Empire that is Ukraine today. Many people therefore wrote Russia in their family trees. So you see here Russia written. But their locations are actually places near Odessa. Oh, welcome. So these are the Black Sea Germans. Most of the immigration happened after 1800 and into a larger area than the Volga Germans. Many evacuated during the 20th century. However, this specific group that we see here still has presence in the Odessa area.
Are you sure it's Germans and not just Jewish people?
Actually, they don't have any Jewish ethnicity.
No?
Almost none. Yeah.
From Stuttgart.
And you can also see their names. The names are
93,000 people have been there in 1940, and then they had to come back because of the Hitler-Stalin Pact. It's at the end of the Danube River into the Black Sea. They came from southwest Germany mostly, and emigrated 1815, 1830 to Russia at that time. Today, Ukraine. So, right.
That is really fascinating. And so now we have a few actually, that describe this migration. So it's really beautiful that this genetic group was distinct enough from the Volga Germans to become a different genetic group. This group is a group with a strong presence here in Amsterdam. I plotted here the distribution of ancestors in 1800 for the 3 most populated countries, and you can see that the Netherlands is leading, and specifically Amsterdam. And it is also really interesting because this group demonstrates an admixture of ethnicities. Looking at the average ethnicity of people in this group, like we've done so far in this talk, on the left, reveals that this group contains many Ashkenazi Jews, but to a lesser degree than typical groups of Ashkenazi that I plotted here on the right. For example, East European Jews or Central European Jews. However, the question remains, are there very little Ashkenazi Jews in this group, or that almost everyone in this group is just a little bit Ashkenazi? So now I would like to talk briefly about the importance of the visual representation of data. This is not only a matter of aesthetics, but also helps us to analyze it more deeply. And I'll give you an example. Let's look at 2 hypothetical groups. Both of them have an average of roughly 25% Ashkenazi ethnicity. But if we look more closely at this representation here, where each individual is a bar, like this one, that is composed from colored components for each of its ethnicities, with the length of the component proportional to the fraction of ethnicity. Using this representation, the group on the right contains 6 individuals, where about 1.5 are mostly Ashkenazi, and the rest aren't. Whereas the group on the left, every person is a combination of all 3 ethnicities. Their average is the same, but this is a completely different group. So let's dive into the admixed Ashkenazi group. On the far left side, we see people who are mostly Ashkenazi, then people who are mostly West European, and they are sorted according to their most dominant ethnicity. Each person is a bar. This plot reveals that while there is a relatively small core of strongly Ashkenazi Jewish that have almost full bars of green ethnicity, most people in this group are admixed Ashkenazi, and probably had at least one grandparent or great-grandparent who was full Ashkenazi 2 or 3 generations ago, that you can see inside these rectangles. So these are the admixed West European Ashkenazi. It's interesting how people from many countries, admixed with many other ethnicities, still clustered with their Jewish relatives. Many still live in Europe. Some of them no longer carry Jewish names. And they come mostly from Amsterdam, Rotterdam, Berlin, and Hamburg. And this is not a large family. These are almost 10,000 people, all coming together into one genetic group. Finally, we reached the last example that I prepared for today. I showed you a number of techniques for data sources that can help us identify the populations within groups. But what if some group is less well documented than others, or is relatively small? So here's a cool trick that we can use to learn more about such groups. We can ask ourselves, what can we learn from the relation between different groups? The matches between them. We can start with the group closest to our center group, the one that has the largest average match between individuals in both groups. In this case, this is a group from Tunisia. I'll just go back one sec and just point out that these two groups, these are two groups shown here on the map, differ genetically, but they are overlapping geographically. And now I'm going to try to discern which is which. So I take one group, and then I look at the closest group to it, and I see that it's still a Tunisian group. And maybe I can't tell more from this neighbor. But I can go on and look at the next neighbor to this group. The next one are Libyan Jews. I already annotated them because I had more data. And now I know that they are Libyan Jews. And that may start hinting at something about the center group. So we continue and add more neighbor groups sorted by their distance to this group. And then we get quite a clear picture of these Tunisians. They are Tunisian Jews with many interactions between other North African Jews and Ashkenazi Jews. What about the other group? So the other group has more matches with other Muslim North African groups, Moroccan, Algerian, and Tunisian. So we can actually learn a lot without any additional information on surnames, ethnicities, family trees. And this helps us to annotate additional groups. So I would like to stop here and summarize the main things that we saw today. Today, I've shared with you some of the work and challenges towards better ethnicity models that we at the science team in MyHeritage are dealing with. We use the DNA samples from our millions of users to process their matches and create various new tools to analyze this worldwide DNA web, mapping this web, if you will, like explorers of ancient times. These groups show us the history of worldwide populations in the past 200 to 300 years through these discoveries of the fascinating mix of segregation, assimilation, and migration. We can see the complex journey humanity is making in defining one of its key concepts, identity. Examining these patterns that made us who we are, we see how similar we are to one another, even though each group is unique and special. We are currently working hard towards bringing these genetic groups to you. And one last thing, if you belong to or can share the story of an interesting population in your country, I would love to hear about it. Come talk to me after the lecture. You are welcome to ask questions, and I'd be very happy to hear your stories. Thank you very, very much from myself and the MyHeritage science team. I marked the main players here that did all the work that I presented. In fact, I'd like to add one more person to this list. Gilad, our CEO, was actually personally very much involved in the research of many of the genetic groups that we detected, and some of them you've seen here throughout the project. Thank you very much, and with that, I'll be happy to take questions. Yes?
Thank you very much. Is it on? Is it? Okay. So thanks very much for the presentation. So as you might have heard yesterday, next door in the Netherlands, over 1 million people out of 17 million have a East Asian and Suriname heritage. So they're admixed. So the question is, and so their ethnicity estimation of their East Asian part is usually terrible.
Yeah.
So my question is, what can you do with the genetic groups for that ethnicity estimation, knowing that this admixed part is very diverse? It's not like Ashkenazi Jews. It comes from a region that's as large as Europe.
So, if I understand correctly your question, you were asking whether genetic groups can be used to fix these?
It can be constructed somehow.
That's a very interesting concept. We haven't started working on that, but this is an interesting idea. I can't tell you off the top of my head how many such genetic groups we have, if any, of Suriname people in the Netherlands.
Or Dutch East Asia especially.
Yeah.
Because the Suriname, of course, they trace back to Africa and also to East Asia.
But-
But I mean, this is a source of big frustration for this, 1 million people in principle.
Yeah.
Because this East Asian, the interest or one of the interesting parts of their DNA, their ethnicity estimates are usually way off.
So I do hope that with genetic groups, as the first step, we will be able to give these people better estimates in the form of associating them with Southeast Asia genetic groups. And we do have Southeast Asian genetic groups. I don't know about these specific populations, but for sure, the genetic groups are giving us a better granularity and also a worldwide spectrum of ethnicities that we can associate people to.
The problem is that there's very few groups endogenous groups within Indonesia that are tested. I mean, that's a challenge.
Yeah, but I do hope that we have enough Indonesian people that actually bought our kits, either in Indonesia or in the Netherlands. I do hope. Yeah. Well, I do hope that, like the admixed groups of Jewish people, we will find admixed groups of other ethnicities as well. But I can't promise that until we actually...
research, you should construct a genetic group from these people.
From the Netherlands, from the Netherlands. You should take out the parts, the Asian parts
Yeah.
of the Dutch population and create, based on that, Asian subgroups.
That's a great idea. That's a great idea, especially if you can help me and bring Indonesian people from the Netherlands to take the DNA kit.
There are many, there are 1 million.
Yeah. Yes.
All your examples were from Europe. Do you have any genetic groups in Africa or Asia, and India particularly?
So I can't give you exact numbers, but we do. We do have all over the world. In Latin America, in North Africa, you saw the Tunisian groups and the rest of North Africa. And it still remains to be seen once we research all of the hundreds or thousands of groups that we have and see how they are distributed. So these are only first examples, like a small taste of what we can achieve, in my opinion. Yeah, at the back.
Do you filter out the Jewish endogamy for false positives in your extended generations?
Can you repeat that?
Filter out the Jewish endogamy in the extended generations of all the matches in MyHeritage?
No, so not explicitly, no. That's a good question.
It would bring down your matches considerably.
Yeah, but at this point we don't do that. Yes.
Yeah, hello. I'm sorry for my English, but I'm from Barcelona, from the Iberian Peninsula, and I'm interested to know if the Basque people have an own ethnicity, and if thanks to that, anybody knows where they did come from.
So I hope so. There was another question here, right? Yeah.
Thank you. My sister-in-law was surprised by my heritage, that she was 50% English. And we know that nor she or her ancestors even visited the UK. So I presume the Anglo-Saxons were her ancestors, and in the UK they have descendants too. I think for you it will be increasingly problematic when German people join MyHeritage, and you write them that they descend of that they are 50% English.
Yeah.
So maybe that's something to look about.
That's definitely something that we are aware of and trying to constantly build better models. I hope that more models will come, not only genetic groups in the future. And I'm sure that any upgrade will improve these ethnicity estimates.
I hope so. Yes.
Thank you.
Thank you.
Okay, so thank you very much. Please come see me later if you have more stories to share. Thank you.